if that's okay.
We're happy to introduce Rick Stevens from University of Chicago and Argonne, who will give this wonderful talk about the systems biology knowledge base.
Thanks, Jeffrey. Sorry I couldn't be there in person. I'm in Park City, Utah at a workshop. So I'm going to talk about a project that we've been working on for quite a while. That recently got a big boost in funding called the DOE Systems Biology Knowledge Base. So I'm going to skip through the first slides. I have some pictures of Chicago in here and Argonne, just so you know where I'm coming from. We do a lot of large-scale computing at Argonne. Although this talk does use some of the large-scale machines, it's the project that we're building is more oriented towards distributed systems and cloud backends. We recently installed a new supercomputer from IBM called Mira. It's number 3 on the global top 500 list. There's a picture of it. I'm not going to talk about it. It's just got lots of cores and lots of RAM. It's sort of fun. What I'm going to talk about, though, is the environment that we're building to enable distributed analysis of genomes. This picture I'm showing right now is of a large number of sequencers from a recent visit I took to the BGI in Shenzhen, China, the Beijing Genomics Institute, or what used to be called that. And these are the big hardware for genomics. Each of these machines is capable of producing hundreds of gigabytes of output per run. BGI has a very large number of these machines. They're producing right now about 6 to 10 terabytes of data per day. In fact, a number of the people from BGI are actually at this meeting I'm at here, and they were discussing the ever-increasing amounts of data and the large amount of computing hardware needed just to process the kind of initial processing of the sequencing data that's coming off of these machines. This is the future for genomics. These machines can sequence, if they ran all of them, they could produce tens of thousands of human genome equivalents per year, but they're mainly being used to go after large-scale projects in plant and animal genomes and in microbial genomes, and we're getting an incredible wealth of data being generated from these projects. Now, the reason I'm interested in these problems in biology is that, first of all, biology is probably the biggest scientific discipline on the planet. It has really big questions still yet to be answered. We really don't know whether we understand all the life forms on Earth. We don't really understand the role that life plays in kind of the metabolism of Earth, that is the flows of chemicals, of chemical compounds and energy in the planet, in the biosphere. We have a pretty good understanding of how cells work, but not a complete understanding, and that's still open. We don't really understand how evolution sort of developed sort of the engineering principles of life and so on. So these are some of the really basic questions that motivate what we do. One of the reasons that biology is interesting to me is it's fundamentally different from working in the physical sciences. And the reason for that is that in addition to biological systems having to follow all the laws of physics, because nothing can violate those, there's additional sort of laws or rules that they have to follow that really differentiate them from physical systems. For example, biological entities are living entities, and they have an unbroken chain of existence all the way back to the original life forms on Earth. And in fact, every organism that exists today has in its genetic code some aspect of that history over 3.5 billion years. And that means that to study biology, you really have to study this kind of data-rich, irreversible, time-variant system. And that's what makes it quite different, say, from studying, say, chemistry, where the there really isn't deep history in the chemical compounds that exist. I mean, you might have some things in geology where you've got some sense of history, but in biology it's very data rich. Every organism has a historical lineage, and in order to understand biological systems, we have to build systems that allow us to explore that lineage. If we go beyond individual organisms to study collections of organisms in the environment or social structures, like in humans, then there's even more rules that get built on top of that to really understand that. These kind of higher order principles that govern the interaction of collections of autonomous entities, and those we think of as ecological rules or economics rules or social rules. And this kind of dichotomy was first articulated by Ernst Mayr, who I have a picture here, who's a evolutionary biologist who worked at Harvard for many years. So I can't see everybody who's watching this talk, but I'll assume that you have some basic knowledge of biology. The picture I'm showing right now is sort of the canonical story that we have, that we tell, that we teach. It's also known as the central dogma in biology, and basically, the highest level concept here is the phenotype, that is what an organism is able to do or what it looks like, is a property of that organism's genome plus that organism's environment. And the genome by itself is simply an information storage mechanism that's in the cell, the DNA molecule, but it plays a very dynamic role in how the phenotype actually emerges from that storage. First of all, you've got transcription, copying short reads off of the DNA into RNA that gets translated into proteins. Proteins assemble into networks to build biochemical circuits, and those circuits execute, just like a computer program does, to create phenotypes. And of course, proteins are the machines that actually enable transcription and translation, so there's feedback loops. And on the right-hand side of this diagram are the typical kinds of assays or experimental techniques that we can use to get at what's happening in the cell. So of course we can sequence DNA, we can use microarrays or DNA sequencing devices to get at what genes are being expressed. We can use proteomics technology to understand what proteins are present in the cell at any given moment. We can use mass spectrometry and other techniques to look at the small molecules, the sort of currency that circulates inside these biochemical networks, and we can get snapshots of that. And by putting all of that information together in an appropriate computing environment, we can sort of reverse engineer what's going on in the cell. And in some sense, this is the macroscopic agenda for systems biology, is reverse engineering the systems that make up biological entities. And of course, as a computer scientist or as a technologist, we want to build the tools that enable us to do this very quickly. Now, we're not just interested in individual cells, but we're interested in the whole hierarchy of life that's needed or that exists, right? So on the lower left here, we have a genome represented by this circle. Most bacterial genomes are circular. The next diagram here represents the networks implemented by that genome. Then down to the bottom, this is a electron micrograph of a cyanobacteria. Gives you some sense of the scale. That cell's about a micron or so long, a couple microns long and wide. This particular one is a cyanobacteria, lives in the ocean, and has about 3,000 different genes in it. Those organisms form populations because they don't live by themselves. They form large collections of cells, and then those populations of individual types of organisms join with many other kinds of organisms to create communities. This colorful thing on the upper right is a Winogradsky column that might have 24 different kinds of organisms sorted by their tolerance to oxygen. And then on the lower right is really real environments where any given environment